All right, let's jump right into it. Welcome, focused learners, to today's explainer. I am absolutely thrilled you're here with us because today we're looking at the actual nuts and bolts mechanics of how genes determine our physical characteristics. If you've ever found yourself wondering why you look the way you do, or how incredible human diversity actually operates down on a cellular level, well, you are in the exact right place. We are going to decode the literal blueprints of life by carefully breaking down the source material together. Let's do this.
So we have to start with a really fundamental question. Is human genetics really just dominant versus recessive? You know, most of us were taught basic Mendelian genetics in early biology classes. That classic, almost too simple idea that one allele is strictly dominant and just totally overpowers a recessive one. But nature, and honestly our own bodies, are so much more intricate than that. Simple dominant-recessive interactions are really just the tip of the iceberg, right? And if we stop there, we completely miss out on the incredible mechanisms that actually drive human diversity.
Here is our quick roadmap for this explainer. We'll be breaking down 5 non-Mendelian patterns of inheritance in a really structured, logical way. We're going to cover 1. Beyond simple dominant genetics. 2. Blending versus simultaneous expression. 3. The multiple allele system. 4. Pleiotropy versus polygenic inheritance. And finally, number 5, the complexity of our traits. Okay, moving right into section 1, beyond simple dominant genetics, or deviations from Mendelian rules. Which naturally brings us to section 2, blending versus simultaneous expression, or allele versus allele. We're going to start off by looking at what happens at the micro level, when 2 distinct alleles just completely ignore those classic rules of dominance. Let's draw some clear boundary lines between our first 2 concepts here. On one hand, we have incomplete dominance. This is where neither allele is completely dominant, which results in this intermediate or essentially blended phenotype in a heterozygous individual. But then on the other hand, we have codominance. This is where both alleles are simultaneously and fully expressed in the heterozygote without any blending whatsoever. So it's not compromise, it's a literal coexistence. And honestly, this brilliantly illustrates the difference. Look at this snapdragon shown here. When you cross a red-flowered parent with a white-flowered parent, you don't get red or white. You get an entirely new intermediate phenotype in that first generation, a pink snapdragon. It's exactly like mixing paint. And if you cross those pink flowers, you get a 1 to 2 to 1 phenotypic ratio of red, pink, and white. That right there is incomplete dominance, a true blend. Now, contrast that with the roan flower. When you cross the red and white parents here, the offspring doesn't blend into some pale pink. Instead, you see patches of pure red and patches of pure white completely coexisting on the very same flower. Both of those alleles are basically standing up and saying, nope, I am going to be fully expressed.
That takes us to section 3, the multiple alleles system, which I like to call the crowded gene.
So what happens when a single gene has 3 or more alternative alleles just floating around in a population? I mean, most genes we think about exist in just 2 forms, but biology loves to give us options. And the perfect example of this is literally flowing right inside our own veins. Using the human ABO blood group system, we can see it's actually governed by 3 distinct alleles in the human population: IA, IB, and little I. You can see the various combinations we've mapped out here. But remember, even though there are 3 options available in the overall population, any single individual can still only inherit a maximum of 2, 1 from each parent. And it's the specific combinations of these 3 alleles that dictate whether you have type A, type B, type AB, or type O blood. So the absolutely crucial point here is that human ABO blood types actually exhibit 2 of our non-Mendelian concepts at the exact same time. It's wild. They show both multiple allelism and codominance. Think about it. If you inherit the IA allele from one parent and the IB allele from the other, your genotype is IA-IB, which gives you type AB blood. Neither A nor B is dominating the other. Instead, they're completely codominant, meaning a person with type AB blood fully expresses both A and B antigens on the surface of their red blood cells, all at once.
All right, now we tackle our final concepts in section 4, pleiotropy versus polygenic inheritance, which we're calling the Great Reversal.
We are basically pitting 2 massive genetic ideas against each other right now, the multitasker gene versus the team effort. First up, let's look at the multitasker, pleiotropy. This wild phenomenon where a single gene influences multiple, seemingly completely unrelated phenotypic traits. A rule of thumb, well, actually, scratch that. It's more like a mnemonic anchor, is simply one gene, many traits. It's fascinating because it really highlights how one localized tiny genetic change can have massive, widespread, cascading effects across an entire organism. Let's ground this with a real medical example so you can see what I mean. Phenylketonuria, or PKU, is an autosomal recessive disorder, and it's caused by a mutation in just one single gene, the gene responsible for the enzyme phenylalanine hydroxylase. But just look at the varied, widespread effects this has. Because of that one single mutated gene, a patient can experience a huge variety of seemingly unrelated symptoms, ranging from intellectual disability all the way to reduced hair pigmentation to skin conditions like eczema. That is one gene acting as a massive multitasker, causing all these different effects. Now, what is truly fascinating is comparing these two exact mechanisms side by side. On one side, we have the one gene to multiple traits pathway of pleiotropy, using sickle cell anemia as our model. And right next to it, we have the exact opposite mechanism, the many genes to one trait pathway of polygenic inheritance, using human skin color as the model. Let's look a bit closer at the mechanics of both sides. Zooming in on the pleiotropy side, let's look at sickle cell anemia. A single mutation occurs in the HBB gene. That's it, one single gene. But that one mutation causes red blood cells to become sickle-shaped. And then that one physical change, this devastating domino effect, occurs throughout the entire human body. It leads to anemia, it causes blockages in the blood vessels, which in turn leads to severe fatigue, an enlarged spleen, and immense bone pain and damage. It's such a powerful reminder of how interconnected our biology really is. You pull one single genetic thread out of place, and it unravels multiple parts of the whole biological tapestry. But then, flipping the script entirely, we have polygenic inheritance. This is where one single trait is controlled by two or more genes working together. So our rule of thumb here is many genes, one trait. Instead of a single gene acting as a bottleneck for many traits, we have a whole team of genes pooling their influence together to dial in just a single physical characteristic. And because each individual gene in this team adds just a tiny cumulative effect, kind of like dropping tiny drops of paint into a bucket, we don't see harsh binary outcomes. Instead, we get this continuous, incredibly wide range of variation in populations. It typically results in this really elegant bell curve distribution. And it perfectly explains the beautiful, continuous spectrum of human skin color, eye color, and even our height. There isn't just tall or short or light or dark. There is an endless cumulative gradient, and it's all determined by multiple genes working in concert. Which brings us to our final section, the complexity of our traits, synthesizing human diversity. So we've looked at the microscopic mechanisms, and now it's time to step back and look at the whole picture. To summarize what we've unpacked today in this explainer, these 5 deviations from Mendelian genetics, they are truly the architects of our biological nuance. Incomplete dominance gives us those blended intermediate traits. Codominance allows traits to proudly coexist without having to compromise. Multiple allelism expands the entire menu of genetic options in our population. Pleiotropy shows us just how deeply interconnected our biological systems are to single genetic anchors. And polygenic inheritance paints our traits with the fine, continuous brushstrokes of multiple genes. Simple dominant and recessive interactions gave us the foundation, sure, but these 5 patterns, they give us the real complexity of life. Before we wrap up, I want to leave you with a thought to apply immediately to your own life. Next time you look in the mirror, really consider your own height, the exact unique shade of your eye color, and even think about your blood type. Which of these 5 complex genetic patterns made you who you are today? Because you are literally a walking, breathing masterpiece of non-Mendelian genetics. Thank you so much for joining me on this explainer today, focused learners. Keep asking questions, keep studying those blueprints, and I'll see you next time.